All right, so this will be your study guide to the multiple choice part of the test. Scientific method. Know the steps, their definitions, and the correct order. So for the steps and the definitions, you might have like a short sentence where it says, Mrs. Compton thinks that if you spend 3 hours studying for the test, then your test scores will increase. What is the dependent variable? So in that situation, I'm measuring your test scores increase. So the dependent variable is your test scores increase. Your independent variable is that you study for 3 hours. How much time are you studying? And then my hypothesis is I think if you studied for 3 hours, then your test scores would increase. My question would be, does student time studying affect their scores on the test? So the correct order of the scientific method is observation, then question, then hypothesis, experiment, results, analysis, conclusion. Theory versus a hypothesis. What is the difference? And be able to identify one versus the other. So I'm trying to look on your main test to find the question of the theory versus a hypothesis. So a scientific theory is something that is surrounded by scientific evidence. It's supported in evidence. It's got years and years of evidence to back it up. A scientific hypothesis is something that you think might happen and that you can test individually. So your test question asks you to identify which one of these is a scientific theory and not a scientific hypothesis. So again, you've got to look at a big overarching idea for a theory that's got years and years and multiple evidences to back it up. And a hypothesis could be something that you could come up with and you could test on your own. Structure and function of organelles. There are quite a few organelles that you kind of need to know the structure and function of, like the mitochondria. It's the powerhouse of the cell. Of course, you know that, but it is where the cells make their energy. The endoplasmic reticulum is what modifies proteins. The Golgi apparatus is responsible for the modification of the proteins that the endoplasmic reticulum helps to, again, modify, and the Golgi apparatus will modify it further and package it for use in the rest of the cell. The ribosomes are involved in protein synthesis as well. They are actually what puts the amino acids together. So ribosomes make it, endoplasmic reticulum modifies it, the Golgi apparatus will package it and modify it further. So your question might ask you which of the following is not a part of protein packaging and synthesis. So you'll have to know at least those three are the main components in that. Parts of an atom. The parts of an atom are the protons, they are found in the nucleus, the neutrons, which are found in the nucleus, and the electrons, which are found in a cloud surrounding the nucleus. Definition of an inert gas. An inert gas is not going to bond with anything else because it already has a full outer shell. Isotopes. How do you calculate numbers of neutrons? So, for example, you have carbon-12 and carbon-14. Carbon-12 has 6 protons, 6 neutrons, and 6 electrons. And you get that because 6 plus 6 gives you 12, and 12 is that identifier at the end of the number. It's the mass number. Carbon-14, it's got a mass of 14. So you know that carbon has 6 protons because its atomic number is 6. So you take 14 minus 6, and that gives you 8. So carbon-14 has actually 8 neutrons, and the number of protons is always going to equal the number of electrons. So carbon-14 will also have 6 electrons as well. Properties of water: adhesion, cohesion, density, solvent, pH, heat of vaporization. That's a lot. Adhesion is where water sticks to another surface. Cohesion is water sticking to itself due to its hydrogen bonding in between the water molecules, the negative oxygen and the positive hydrogen. Density: water is actually less dense as a solid, which is a unique characterization because the hydrogen bonding forming a lattice between the water molecules as it freezes, and water that is just before freezing, about 4 degrees Celsius, is actually going to be more dense and sink to the bottom, which is how fish stay alive. Water is called a universal solvent because anything that's polar or ionic dissolves in it. Water in its pure form has a neutral pH, but it can be acidic or basic depending on what's mixed in it and how it works. And heat of vaporization, it's got a high heat of vaporization, meaning it takes a lot of heat to get it to move from a liquid form to a gas form. Acidic solutions versus basic solutions in terms of hydroxide and hydrogen ions. An acidic solution is going to have greater hydrogen ions. A basic solution will have more hydroxide ions. Characteristics of ionic, polar, covalent, nonpolar covalent, and hydrogen bonding. So this is actually a matching question on your test. And you have to match the characteristics of those chemical bonds with the types of bonds. So the bonds within a water molecule, those are hydrogen bonds. Ionic bonds are bonds that would readily fall apart in water. They very quickly fall apart because they are magnets basically, and they will be attracted to the different parts of the water molecules. And it's also a bond that holds sodium and chlorine together. And it's a bond where atoms will transfer electrons. Your polar covalent and your nonpolar covalent are both bonds where electrons are shared. However, in the nonpolar covalent, the electrons are shared equally. I don't know if you can hear that in the background, but my baby is screaming. And in the polar covalent, it's where the electrons are shared unequally. And the bonds within a water molecule itself are the polar covalent bonds. The bonds in between the water molecules that give its cohesiveness, that's the hydrogen bonds. Those aren't really true bonds. And a bond in between two water molecules, that's a nonpolar covalent as well. So your next one, characteristics of the 4 types of biological molecules. This is also a matching question. So you've got proteins, carbohydrates, nucleic acids, lipids, or you will have a choice of all 4 of the above answers. So long-term energy storage, that's a lipid. Lipids also includes waxes or oils or fats, and they're for waterproofing purposes. Carbohydrates include sugars, anything that ends in O's like fructose, glucose, lactose. Those are all sugars, and those are carbohydrates. Carbohydrates also include things like cellulose and starches, which are polymers, polysaccharides. You have nucleic acids, and those include your nucleotides. Your nucleic acids are your nucleotides. Those are the little A, T, C, and G that makes up DNA. You have your proteins, and proteins are made of amino acids. Those are the building blocks of your proteins. And they also have peptide bonds in between them. But they all contain carbon. That is the one thing that they all have in common. Okay. Definition of organic molecule. An organic molecule has carbon to hydrogen bonds, and it's used in life. And so if I would say like CH3, that's an organic molecule. If I would say C6H12O6, that's an organic molecule. If I would say like CO2, that's not an organic molecule. CS2, that's not an organic molecule. Trying to think of other examples. CCl4, that would not be an organic molecule. However, I don't know if that's a real thing that would actually bond. But it has to have that carbon to hydrogen bond. No dehydration synthesis and hydrolysis and how they work. So at dehydration synthesis, we are removing water. It's also called a condensation reaction. So you're taking water out of 2 separate molecules and joining them together. So you're building a polymer that way. Hydrolysis, you're using water and you're coming into a polymer and you're going to add water and it's going to break apart polymers into 2 separate sections, whether they be monomers or like oligosaccharides or something like that. They're just breaking them apart into smaller sections. No parts of a nucleotide, lipid, carbohydrate, and protein. So there's a lot there. Proteins have your amino and your carboxyl groups. That's how you can tell the amino acids that make them up. Carbohydrates are made of monosaccharides, and they are carbon rings that build those. Lipids always have a fatty acid chain, a carbon to hydrogen chain with a carboxyl end, and it's attached to some kind of like a phosphate or a glycerol group, something on the end of it that holds these long chains of carbons and hydrogens. And a nucleotide has a nitrogenous base. Always has some kind of nitrogen involved in it, and a phosphate group and a monosaccharide carbon ring of some sort. Know how peptide bonds form. So the carboxyl group of one amino acid will join with the amino group of another amino acid and make dehydration synthesis occur. Water comes off and they will join together. Yep, that's basically it. I skipped one, is what I was looking at, sorry. It says, note the following groups on molecules. So a hydroxide group versus a hydroxyl and amine group. Hydroxide is an OH negative. A hydroxyl group is an O bond H attached to something. Amine group is NH3 plus or NH2. Carboxyl is C double bond O bond OH. A hydrocarbon is a bunch of carbons and hydrogens joined together in a chain. And R groups are the functional groups off of amino acids. They come off of it, and that's what determines how that amino acid is going to act in the tertiary structure of proteins. Basic structure and point of functionality on the level of organization of proteins. So a primary structure is a chain of amino acids joined together. The secondary structure is how those hydrogen bonds in between the amino acids. It's going to cause it to form like a spiral or a zigzag chain. The tertiary structure is when the R groups start interacting with each other in that spiral or zigzag, and they kind of cause it to fold in and among itself and make different bonds, and that's when it's considered functional, is at that tertiary structure. And then sometimes you have even more complex at the quaternary structure, where you have a few tertiary structures that join together. Those are quaternary structures. Hierarchical organization from cells to biosphere. So more simple than you need to know, but atoms make up molecules, molecules make up organelles, organelles make up cells, and cells are considered alive. They're the smallest unit of life. Cells make up tissues, tissues make up organs, organs make up organ systems make up organisms, organisms make up populations, populations, groups of those together make up communities. A population is a single species, a community is more than one species of living things. Communities make up, or communities and their non-living components, I should say, make up the ecosystems. Multiple ecosystems together make up your biomes, and multiple biomes together make up biospheres. Cells theory, 3 parts. So all living cells arise from pre-existing cells. Cells are the basic units of life, and all living organisms are made of cells. 3 parts to your cell theory. The endosymbiotic theory says that a long time ago, prokaryotic cells joined with other cells, and eventually they started living inside of them through symbiosis, and they gave rise to mitochondria and chloroplasts. And it says 2 major types of cells are. 2 major types of cells are prokaryotic, which are cells that do not have a nucleus, and eukaryotic, which are cells that do have a nucleus. So I went over most of the matching already, except for the cell parts, the types of cells. So all cells have DNA. All cells have ribosomes. Only plant cells have a large central vacuole. Only eukaryotic cells have endoplasmic reticulum and mitochondria. So eukaryotic cells include protists, fungus, animals, and plants. So there's your study guide notes for the multiple choice section. I pretty much went over the test with you. I really hope you have a successful time studying, and we'll see how it turns out when you take your test. See you this week. Bye.